Forget frequently asked questions.
Common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
Go above and beyond.
they become very good at what they do, but only
0.1%.
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
With Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius Podcast. I have Gareth Brady. He's an Ussher Assistant Professor in Clinical Medicine at Trinity College, Dublin. A beautiful place. I walked the grounds in Trinity College once before, and it's really nice. So today we're going to talk about, he's going to answer some of the questions for the virus book that I'm working on. But his current work is Translational Information Research Group, TIRG. They're investigating how viruses target and inhibit common pathways that drive inflammation in human cells, and then they use the knowledge to develop novel strategies to therapeutically inhibit these pathways. So that's like a real basic overview, but welcome, Gareth. How you doing?
Good. Thank you very much for inviting me. Pleasure to be here.
Yeah, in a better way, tell me about your research in your words. It'll come out better, and what's it about?
Sure. So my interest is in studying viruses that are very efficiently able to target innate immunity. And so all viruses develop some skills in doing this, but some are better than others. And so I did my PhD on vaccinia virus, which is a pox virus, or was the vaccine strain used to eradicate variola, the causative agent of smallpox. But during my PhD, it became quite clear that it wasn't a great virus to study to try and find out how to turn off human pathways, because manifestly this virus doesn't do it, because that's why it works such a good vaccine strain, because it drives the immune system nuts. But there is one virus that's specifically adapted to human infection. It's the only virus we know of that does this, and it's called molluscum contagiosum virus. And it only infects humans, and it's way out on its own in the family tree of pox viruses, and it's completely different from vaccinia virus, and very, very understudied before I started working on it, because, of course, the most interesting thing about it is it doesn't cause disease. And that, on the one hand, makes it a very, very interesting thing to study, because if it doesn't cause disease, then that clearly indicates that it's very good at getting around the immune system, because most virus disease is caused by the reaction against the virus by the immune system rather than what the virus is actually doing itself. But that, unfortunately, also makes it kind of a funding liability, because, you go to a funding body and say, hi, can you give me some money to work on a virus that doesn't cause disease? It's obviously not going to be a very saleable thing. But so my approach really is to try and understand how it goes about evading the immune system and switching off the pathways that we use to detect virus and respond to them. And perhaps, reveal new things about the pathways we didn't know before, because the viruses are evolving things that target specifically the rate limiting steps of these pathways. So they're almost acting like a sort of an investigative probing on the pathways to try and figure out the best bits to inhibit. So if your objective really is to try and find novel ways of switching off pro-inflammatory pathways, then no better way to go about it than studying a virus that's evolved to specifically target the best bits.
Oh, wow.
So that would be, in a nutshell, really, is what the project is on.
Well, quick question here. Of the pathways that viruses are known to inhibit at just the right point, how many steps are in some of these pathways? How complicated are they?
Well, a lot of this has really only emerged in the last 10 to 20 years. I mean, for most of the history of immunology, really, we've known quite a lot about adaptive immunity, because there's two parts to immunity: there's the innate immune system and the adaptive immune system. And the adaptive immune system, I'll start with first, of course, that's all the downstream stuff. That's the T and B cells. The B cells produce antibodies that specifically target peptides in the virus. So that's if you get a new virus infecting, it has lots of proteins, and those proteins are made up of peptides and epitopes. And so you develop antibodies against specific peptides in those proteins. And then you've got T cells that do the same thing, but their job really is to target virally infected cells and kill them. So we've known about that for a very long time, at least about 60 years or possibly even more. But what wasn't known is exactly what kind of triggers it. So for example, if you took a dish full of cells and you infected it with virus, for a long time they knew that a lot of nothing happened for several hours, and suddenly the cells start pumping out very specific soluble factors that are tailored for whatever the pathogen is. So in the case of viruses, the cells start producing lots of what are called interferons and also pro-inflammatory cytokines. And the interferon job is to basically percolate out from that initially infected cell and stimulate all their surrounding cells to turn on about 500 genes. And those 500 genes make those cells not permissive for incoming virus, when the virus then leaves the initial cell and tries to infect those surrounding cells. And then the pro-inflammatory cytokines drive inflammation. And the job of inflammation really is to swell up the area and trigger to the adaptive cells to come in and start responding so that you can generate that very potent adaptive response that's needed to get rid of the virus completely. So what was discovered in the late 80s and sort of early 90s, where there was these specific sensors that were able to sense invariable features of all these pathogens. So the first one that was discovered was by a chap called Bruce Beutler and another guy called Jules Hoffman. And what they discovered was that there was a receptor called toll-like receptor 4. And this was able to detect lipopolysaccharide, which is a feature of gram-negative bacteria. And that effectively is what mostly drives septic shock. And septic shock is one of the biggest killers in the world, you know. So what it does is LPS binds to TLR4, and that drives gene expression, leading to the production of these pro-inflammatory cytokines, and so much of them that it drives what's called systemic inflammation, and you get a very high fever and all kinds of sort of system-wide effects, and then you effectively die as a result. So it's a very, very potent way of driving sepsis. Now, that was the first one that was discovered, and it won the Nobel Prize in 2011 for that discovery, because subsequent to that, they started to go looking for other kinds of sensors that were able to detect, for example, viruses. And so for the last 20 years and 30 years...
One clarifying question there. So can you break down the steps in that mechanism a little bit more?
Yeah.
So it's causing the cell, an infected cell, to express certain, antigens on its cell surface that then triggers a strong immune response, or am I missing it? How does it work specifically?
So what happens is, so they discovered a whole lot of sensors for viruses inside cells. And what these sensors are detecting are the things that virus can't get away from producing. So viruses can mutate everything basically and change all their proteins. But the one thing they can't get away from having to have is nucleic acids to encode these proteins, you know. So we've evolved these sensors for viral nucleic acids. And what they do is, when they bind to these viral nucleic acids, they trigger what's called a signal transduction pathway. And that feeds a signal downstream to what are called transcription factors. And then they jump into the nucleus and bind to loads of genes and switch them on. And the most important genes in that process are these soluble factors I talked about, like interferons and pro-inflammatory cytokines. And both of those, and all of those soluble factors essentially are bringing about all of the effects necessary to trigger the second part of the immune system, which is the adaptive immune response. So what viruses are doing effectively is they're targeting those early steps. So they're stopping the sensors working. They're targeting the signal transduction pathways. They're stopping the transcription factors getting activated. And if they can do that successfully, then they're effectively flying under the radar of the immune system, because we've got to nip it in the bud at that stage before it gets further on to triggering this adaptive response.
Yeah, that's amazing. I guess so my answer is they're able to very specifically surgically interfere in unbelievably complex pathways. Is that one way to put it?
Yeah, I mean, surgical is exactly the right word. I mean, it really is surgical. And one of the reasons why it ends up being surgical is because, particularly pox viruses, is what they're doing is they're actually stealing genes from the body. They're stealing fragments of genes from the body. And they're expressing sort of partial versions of the intracellular proteins, and they're using these almost like decoys. So for example, if you can imagine there's a protein in one of these signal transduction pathways that transmits a signal. Well, if the virus steals part of the gene for that protein, it then expresses it, and then that fragment of it is able to decoy the interaction between proteins in that pathway and completely stop it transmitting the signal downstream. So they're doing this all the time. They're picking up gene sequences, they're trying them out, they're getting rid of what doesn't work, they're taking up new things. And you can imagine over, you know, thousands or millions of years of doing this, they get extremely surgical about specifically targeting the best bits. They don't bother targeting anything that's not effective or efficient. They're only going after the most important bits. And that has been, we've shown that time and time again, that, you know, it's almost predictable in a sense what bits of the pathway they're going to target, because you can see upstream there's no point in targeting multiple pathways that are all ending up activating a common core pathway part downstream, if you know what I mean. So they'll just go after the bit that's common to all the upstream bits. So it's quite surgical. I mean, it's a very good way of putting it.
So this was one of the questions I had. if you consider a pathway and all the elements of it and all the steps in it as an information space, and viruses are essentially exploring that information space and figuring out targets and then, interfering in a, like I put it, a very surgical way. Do you think that viruses could be harnessed like biological supercomputers or like biological AI to explore other information spaces? For instance, if a given virus fuses to and enters a cell, could you somehow create an experiment where viruses, by changing the let's say the receptors on the cell surface, et cetera, they'd be forced to explore that information space and figure out novel ways to enter the cell that we couldn't do without tons of grunt work? Do you think they could be used in that way?
Well, and that's precisely how we use them in a sense, because we're using them as sort of microcosms of evolution in a sense, So if you allow a virus, particularly an RNA virus, actually is a better example, because RNA viruses evolve very rapidly because their genomes don't copy with the same fidelity as DNA viruses like pox viruses do. So they evolve tremendously fast, So in a sense, the end product of the evolution of a virus inside a particular host is like that experiment running, So again, taking the example of molluscum contagiosum, when you observe the lesions on the skin that it forms, these lesions are completely chock full of virus. They're loaded full of virus, and it can last for years, and it's very, very unusual because most viruses that last for years go into some sort of latency or dormancy. They hide from the immune system by switching off gene expression or deposit themselves in our genomes and just hide. But molluscum just stays there for often years. It can last up to 4 years in some cases, and it's always producing all of its proteins, and it's always present. And yet the lesions are never inflamed. They largely persist as if the immune system doesn't even know it's there. So in a sense, when you look at that system and you say, well, that's impossible, because we know that the skin particularly is the vanguard of the immune system. The keratinocytes in the skin express all of the sensors I was talking about. Some cells express some of the sensors, other cells express other of the sensors. The skin, we now know, expresses all of them. So it's absolutely primed to sense any viruses coming in, particularly molluscum. So in a way, you're looking at it from the end point and saying, look, it's very good at switching off these pathways. We just need to figure out how it's doing it. And if we figure out how it's doing it, then we know exactly what bits to go after in these pathways for switching off human inflammation, for example. And inflammation underlies all human disease, arguably, either directly or indirectly. So
yeah, that's amazing. Do you think that that viruses beneficially contribute to an organism's immunity and immune system, or it's only they're just evading it and messing with it and regulating it for nefarious purposes? Or do you think there's ever a beneficial component of it where the virus actually helps the organism's immune system?
The answer is yes, but indirectly. And so the process is really actually like a Cold War and an arms race. The complexity of all these systems in human cells, for example, it's evolved to be complex because viruses have become better and better at evading these pathways. So if you can imagine, if you look in Drosophila, for example, Drosophila has a very reduced, Drosophila is a fruit fly, by the way, so it's a very amenable system for genetic study. So if you look in Drosophila, it's got a very, very reduced set of these sensors and these systems. But if you look at a much more complex organism like humans, it's a massively expanded set of pathways and sensors. And I believe that the reason for that is because the viruses that infect us have been targeting these pathways for so long that it's forced humans to evolve multiple sensors to try and detect them so that they can't easily evade. And at the same time, these viruses are evolving more and more inhibitors to target these pathways. So as the virus is gets better at avoiding the pathways, vertebrates like humans have to develop newer and newer ways of stopping it from slipping under the radar by having, like a burglar breaking into or a diamond thief breaking into a museum or something, and it's got all these tripwires all over the place. If it gets good at avoiding certain tripwires, you've got to design more nefarious ways of triggering him entering in to try and steal the diamond, if you can use that example. And so in a sense, the virus is driving our evolution to get better and better at sensing pathogens. And some of these sensing pathways also have fringe benefits of also detecting bacteria and also detecting cell damage. And one really good example of this has only come about in the last couple of years. There was a protein called C-gas discovered in 2016 by a guy called James Chen. And C-gas detects DNA, but it doesn't specifically detect viral DNA, but it's almost certainly evolved to detect DNA viruses. And we can see that with knockout experiments in mice, that they very, very much have a problem detecting DNA virus specifically when you infect them.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
Now, because C-gas evolved to detect DNA in a non-specific way, in a sense, they're just looking for decompartmentalized DNA. So what's decompartmentalized DNA? Well, most DNA is in the nucleus. Well, all of our DNA is in the nucleus, and some of it's in mitochondria, but none of it's in the cytoplasm. So we've evolved the system to say, well, any DNA in a cytoplasm has got to be a virus. And so we've evolved this system to detect DNA. Now, as it happens, there's also bacteria that can infect cells, like tuberculosis is a good example. And when that gets inside, C-gas is able to detect that. And furthermore, when you damage a cell and the nucleus gets damaged and DNA leaches out, it also triggers that process and also accelerates the process of wound healing. And that's a very, very important job that C-gas does. So the virus has actually sort of benefited us by forcing us to derive these new systems for detecting not only their presence, but also the presence of lots of other things. So it's massively speeding up evolution in that respect by forcing us to adapt constantly to the outside infectious environment.
Yeah, I know in bacteria, the phages the bacteria will take different elements of the phage DNA and or the phage genetic material and incorporate it. I've even heard of bacteria being able to express spike proteins like some viruses have and use it to puncture other bacteria. So it seems like viruses are tools. Viruses use cells as tools. Cells use viruses as tools. What do you think governs who uses who?
I just think it's very much a battle between both, I mean, it's a battle to survive, and the fastest evolving genes in human genetics and indeed, I guess, all animal genetics are innate immune genes. And that's primarily the reason. And you can really see it happening, when certain populations are slightly more resistant to an infection because they've got an abrupt mutation that gives them a preferential survival advantage over another population. And that's an example of that sort of evolution happening in human populations. And there's been a couple of examples of that over the years where that's happening. So, and that doesn't really happen with anything else but innate immune genes, where you can see those kinds of changes happening in human populations in real time almost.
Yeah, it's amazing. Well, one of my earliest questions is, are viruses alive? Why and why not? What's your thoughts?
Yeah, I mean, that's a sort of classic day one virology lecture question. I give a lot of virology lectures myself, and sort of, you kind of end up asking that question, to get the noggin jogging, for students to think about it a bit. But I've always sort of internally thought it was a sort of pointless argument. It's almost like asking the question about is Pluto a planet, I mean, it kind of comes down to the question of what you define life as. Both the alive and non-alive answers are sort of correct in a way, depending on how you define life. I mean, they are alive in the sense that they're replicators. And in some cases, like pox viruses, they're more alive than others, because pox viruses actually look like sort of reduced cells. And it's very, very likely that pox viruses were originally single-celled organisms that found it beneficial to get inside cells and eventually started to lose all the genes that allowed them to live independently. And there's a benefit to doing that, because if you can strip away all the genes you don't need and just use whatever the cell has, then the time to replicate your genome is faster. So you can produce more virus quicker. And then the more virus you can produce, the more infectious you are. And that's an obvious benefit and obvious selection to do something like that. And there's very good evidence for this, because the polymerases, like the DNA polymerase and the RNA polymerases, which facilitate replication of the genome and transcription of its genes respectively, look far more like archaea versions of those polymerases than they do like the host polymerases. So there's quite a bit of evidence to suggest that they were originally single-celled organisms that basically just became dependent on the cell. And another good example of this, of course, is mitochondria. Mitochondria are in every single cell in the body, and we are absolutely dependent on them to make energy. Now, mitochondria There's a very strong theory about mitochondria and also chloroplasts, by the way, the things inside plant cells that facilitate photosynthesis. Both of them have their own genomes, and they're circular genomes. And they look quite like bacterial genomes. And so it's called endosymbiosis. And the idea is that at some point, probably billions of years ago, these single-celled organisms got inside ancestral versions of our cells, and they provided an advantage to the cell because they were able to generate energy. And so the cell just said, okay, why don't you stay here? And then they got rid of all the genes they didn't need, and then they became resident within human cells and, of course, plant cells, respectively, in the case of chloroplasts. And so that's sort of, niche. It's like all life in a sense, because, all life sort of falls into niches of opportunity. And in that sense, it comes back to the question of, well, what is life? Well, I think all of these things are arguably life because they can replicate. I mean, that would be the primary definition. Of course, they can't live independently, but certainly, I mean, if you take, for example, colonies, like if you look at lichen, which is a cyanobacteria, living within a cluster of different fungi. So it's a colony of different organisms. One can photosynthesize, the other can saprophytically degrade material and provide energy from that. And they're inseparable. I mean, they can't live apart. They're kind of colonies that are mutually require each other, sort of a marriage of all these creatures to perform that one sort of living function of everything. So as a broader question of what is life, all of these things fall into, well, what is independent? I mean, nothing is really independent from its environment or independent from some other creature.
The reason why I think this is actually a very important question is that if people say, oh, viruses are not alive, then it closes your mind off, in my opinion, to many things that they could do and why they do it. And if you instead force yourself to say, well, if they are alive, then what? If you look at a given question and you look at it deliberately from both perspectives, I think you'll get very different answers. So that's why I think the question is actually really important. Like here we go. I'm going to ask you some more and think about it from both angles. So, why are some viruses, able to be latent in a host for years or even its lifetime? But, when the host experiences stress or a problem, now they turn lytic and they're like rats leaving the ship and why does that happen? And how is that decision made? Why are some viruses just, pathogenic and lytic and they just come in and, kill cells and other ones are not? And, how can some endogenize? How could you make that choice to endogenize into a host? How could you, so that's my first question is, why do you think that there's this difference in behavior?
Well, the difference in behavior is not so much intentional in the virus. I mean, that's quite clear from a number of viruses. I mean, I spent a good 6 years working on a very interesting, another DNA virus called Epstein-Barr virus, and that's got a lytic and latent stage. What was originally sort of described, these two cycles as lytic and lysogenic, but that more applies to bacteriophage. But it's lytic and latent in mammalian viruses. And Epstein-Barr virus, when it enters first, so it first infects through the mucosal epithelium, and it infects those cells rather sort of poorly or weakly. And then it gets inside the body and starts looking for B cells, because that's a target cell. And when it gets inside the B cells, it drives a massive proliferation in the B cells. So it has loads of different molecules that mimic all of the different signals a B cell needs to become activated. And in the process of doing this, it's also going through lytic replication. Now, lytic replication, by the way, doesn't always lyse the cell. A large part of it is because the virus is going through entry, replication, generating virus factories and producing new virus, which can leave the cell through a very specialized means that doesn't actually lyse the cell. I mean, cells often do eventually lyse because of what the virus is doing to the cell, but that's slightly downstream. Now, what happens is that you're generating an immune response against this virus. And so as you do so, the antibodies are being generated against those lytic genes, the lytic proteins that the virus is producing. And so it puts the virus under pressure to switch those genes off and turn on all of the latent genes, because it's got a whole separate set of latent genes. And what the latent genes are doing are gradually shutting down the virus so that it becomes undetectable from the immune system. And so eventually what happens is that it's only expressing one or two genes, and those two genes produce proteins that just maintain the nucleus, or maintain the genome of the virus inside the nucleus of the B cells. And 90% of the population have EBV genomes inside their B cells floating around the body for life, and it stays there for life. occasionally reactivates, but you generate the same immune response against those lytic genes, and that switches it back to that latent cycle. So it's not a process really of the virus choosing to go lytic or latent. It's actually responding to the pressure of the immune system reacting against the lytic genes, which are more immunogenic than the latent genes.
Okay. When a virus infects, there seems to be a latency period. Sometimes hours, days, weeks, months.
Yep.
Why is that? Do you think it's just exponential growth of the virus and quote unquote enough cells have to be infected, or are there other mechanisms at play? Maybe quorum sensing amongst the virus. Once they're inside infected cells. So are there enough of us? Okay, go.
Unfortunately, they're far too selfish for that. I mean, they operate really as independent entities. There's not a lot of crosstalk between viruses. No obvious crosstalk between them in a way that would benefit any others. I mean, they're operating again quite selfishly. But there's two different sides to that question. The first is, if what you mean is, when do you first start seeing pathogenicity from the virus? Well, the first part of it is how quickly the virus can generate enough of a viral load to drive the immune system to manifest the pathology. And so some viruses, like RNA viruses, divide very quickly. But they do it very efficiently in a sense that they're not suppressing the immune system. So the innate immune system is trying to limit it, and the adaptive response is killing off cells that have virus. However, if they can generate enough of a critical mass of genomes that generates a very potent innate response, then you start getting things like fever, because fever is driven by a cytokine called interleukin-1 beta. And you need quite a lot of that in the serum to generate a fever response, because it actually hits the hypothalamus, and that generates a signal to the liver that tells it to start metabolizing more sugar, and that's what generates the heat that causes fever. So part of the issue is how quickly can the virus generate a critical mass of genomes, of virions and genomes, in order to do this. And the second part of the question is how does the cell respond to the virus? And how does it, the strength of the response from this innate immune response that generates the cytokines that, again, make you feel bad and give you a fever and in some cases ultimately kill you. Now, like I said before about viruses that are very well adapted to the host, they get very good at switching off those innate responses. So that even as they're growing and generating a critical mass of virus, that innate system isn't efficiently triggering the innate response such that it triggers a strong adaptive response to clear it. So it's an interesting interplay between what the virus is doing and how the host is responding to it. And that determines how long it takes before a virus presents. For example, with SARS-CoV-2, the current cause of course COVID-19, and that can take from anything from 2 days to 10 days, and that's probably a very complex dynamic of what that virus is doing inside the host before it generates enough virus to drive an immune response. And what's ultimately killing people, by the way, is the virus seems quite good at switching off the interferon response, so that you generate a lot of virus very, very quickly within cells. But when cells come in and suddenly find all of this material, when they're able to get past that blockade, then they find so much material that it drives a really strong inflammatory response that causes the lung tissue to swell up to the extent, it's called acute respiratory distress syndrome. It's exactly the same as what happens, by the way, in pandemic influenza. And that abrupt encounter of all of this virus material drives a really strong inflammatory response. So it's all these dynamics between what the virus is doing and what the host is doing would be the answer to the question that would determine when you start seeing pathology. And that's what's called the incubation period, obviously. Is the incubation period is not virus-centered, it's more host-centered. It's an observation of what's happening to the host rather than the virus.
Well, along those lines, if there's a virus that makes someone like deathly ill and I get it from them versus of the same virus, but the other person like barely feels anything and I get it from them, do you think I'll my symptoms and I know I'm unique and everything's different about me and all that, my genetics, epigenetics, etc., my immune status, but do you think there's going to be a correlation? If I get virus from a really sick person, will I be very sick, or is there no correlation? Sick or not sick, it's no difference.
There's a very strong correlation, actually, and the correlation is between virus load and exposure. So it was observed very early on in the COVID-19 situation that medical staff in particular were getting very, very serious infections. And it was pinned down, and I mean, it seems there's reasonably good evidence now to show that the amount of virus and what's called the virus load that you're exposed to at the beginning, that you're effectively inoculated with all this virus at once, determines how quickly and how grave the disease eventually becomes. And of course, it makes sense because you've got lots of virus infecting at the same time. It reaches that critical mass faster so that you get a much more dramatic response. Now, that being said, the shedding period during an infection differs between viruses. And so with SARS-CoV-2, people can be tremendously contagious at a very early stage before they even get a spike in fever, and that's one of the reasons why. I mean, there's a couple of reasons why this virus is so contagious, but I would argue that that's probably one of the most important reasons why, because it's dramatically contagious before a person is actually bedbound and sick, for example,
Well, okay, so viral load is a factor that's correlated with probably how sick someone is. Okay. What other factors do you think are important in that interaction? What about my genetic makeup? What about my immune history?
Sure. Yeah.
What about my epigenetic marks? Et cetera.
Sure. Yeah. I mean, look, 80% of people that come across this virus in particular, and I'm sure it's the same for a lot of particularly respiratory viruses. About 80% of people have no symptoms at all, or at least very mild symptoms. They don't require hospitalization. But for about 20% of people, they end up in hospital, and about 3% of those die. And that's a dynamic. Now, the fact of the matter is a huge proponent of it is, of course, immunity of the individual at the time. And there's no doubt about that because of the age discrepancy in the response to this virus. I mean, there's no doubt about it that with every decade after 40, the chance of being hospitalized increases and the chance of dying increases, quite frankly. And that's probably because the immune system begins to knacker out as you get older, like everything else does. I mean, but that's the case for this virus. And if you take the 1918 Spanish flu, that killed mostly younger people. So it's a very virus specific phenomenon who it affects the most and who it doesn't affect at all. But the problem is, all of the data from a lot of these outbreaks is coming from people who get sick. We don't have data on the people who don't get sick because they don't become part of the data, And in the current situation, what you really need to do is we need to go out and start testing everybody for antibodies. And that's one of the things my lab is doing at the moment now. We're developing seroassays for SARS-CoV-2 to try and do population screens, because that's the only way you'll know what the proportion of people who've been exposed haven't really gotten sick at all and then have recovered and have antibodies, compared to the people that become part of the statistics. And then you get a proper statistic on what the infectivity is. So going back to your question about what's the probability if I get exposed to it from a person who's really sick, will I get sick myself? Well, we won't really know that until you have all the data of all of the population, both the asymptomatic people and the symptomatic and, of course, mortally ill people.
Okay. If virions are supposed to be essentially immobile, and they're floating along, and if I know there's a lot of viruses in the world, there's like 10 to the 31 supposedly, and over all time, I don't know how many infections have happened. I'll just a huge number. I don't know, 10 to the 20, let's say. If viruses are so small, let's say 50 to 100 nanometers on average, and their hosts can be microns to meters, how do they find their targets? How do they find the receptor on the cell that's, 10 nanometers in size in this vast expanse of, of potential hosts? How do they do it so many times so reliably without any signaling or, guidance?
Yeah. Well, I mean, the answer to that question is it doesn't really take into account how fast things move, microscopic things move in fluid. I mean, they move dramatically fast. Now, it's driven by tropism for the entry receptor. So if you imagine, for example, HIV getting into the bloodstream through fragile tissue or broken skin in the genitals, for example. Now, it has to go quite a distance before it comes across a CD4 and CD4 positive or CCR5 positive cell, because it's looking for both those receptors, right? And they just both happen to be expressed on CD4 positive T cells. Now, they're rolling around they're floating around this fluid all the time at a very, very fast speed, but so are these T cells. And so what's happening is when they come into proximity, the interaction between the virus receptor and the CD4 and CCR5 molecules is electrostatic. So it's acting like a magnet. So as soon as it comes into a reasonable proximity, it's going to, so electrostatic interactions, it's more physics really, but they decrease with the square of the distance between the two things interacting. However, there's still a kind of a magnet-like attraction between those two things, because they kind of want to find each other from a molecular point of view. So it doesn't take too much time of rapidly moving around in fluid. For example, in the bloodstream, I mean, the virus is going to be moving around throughout the whole body. It's being pumped by the heart, the blood's going around and percolating through the tissue. It's not going to be too long before that finds those cells. And once it does, then it starts to replicate and produce 10 to the 6 versions of virus per 50 cells, whatever. And when it does that, you've just got a huge viral load that then doesn't have any trouble finding any of these cells. But obviously, I absolutely appreciate what you're saying. There is a lag period between entry and finding the target cell, but it's not really as long as you think, because of the nature of how fast microscopic things move in fluid.
Okay.
I mean, it's not the virus itself moving, by the way, of course, it's in fluid motions pushing things around. And you can even really see this under a microscope. The smaller things are, the faster they move, like on a film on a microscope, or on a microscope slide. It's not motility per se, but it's fluid motions.
Okay, gotcha. When someone's infected with a virus, I would guess they're not just infected with one specific genetic sequence. The virus will have, I guess, what they call quasi-species or variants, close variants. So,
yep.
I mean, this is my guess, is when someone's infected by a given virus, so that there's dozens or hundreds or thousands or even millions of variants, slight variants. I don't know if you can call these quasi-species, but do you think that they all coordinate to infect, or there are instances where someone has created an isolate of a virus with just one sequence, and there's no variations, and infected a mouse or a cell or a bacteria, and seen the difference between that and like a wild type that has a bunch of variants?
Yeah, I mean, co-infection is entirely possible. In a lab, you can infect a cell with multiple different viruses. It's called superinfection in the lab, but it's also been observed in patients, and it's called co-infections. You can get co-infections all the time. And so you're talking about variants of one virus, or multiple different viruses at the same time?
Well, no, I mean the same virus. The XYZ virus. Let's say it's an RNA virus, so it has a lot of variants and it mutates a lot. So if I get infected by the XYZ virus, I'm probably not going to be infected by, let's say it has 30,000 base pairs. All 30,000 base pairs won't be the same in all the viruses of the XYZ virus that infect me. It'll probably have like tons of variants. Maybe one base pair is different, or 1,000 base pairs are different, whatever. But, I mean, I don't know if you can call these quasi-species, but they're just closed variants of what we call the XYZ virus. I'm making this up.
Yeah.
So when I get infected, do you think that these variants coordinate and they have varying abilities to be able to infect me better than just one, very homogeneous strain that's exactly the same?
Yeah. There aren't as many variants as you would think. I mean, so looking at the sequencing studies from SARS-CoV-2, right, in the current outbreak. So when you sequence the virus, and of course with next-gen sequencing now, it's trivial to sequence the entire thing in a very short space of time. You just stick it into the machine and it will just give you back a sequence. It's not like the old school Sanger sequencing, which took, days or weeks to walk along the chromosome, or the viral DNA. Like it's trivial now. So they've done this all across the board. And what you would typically expect to see in these viruses is about one base pair change per month. So it's not that fast as you would think. And one of the reasons is, because going back to the RNA virus making mistakes all the time when it copies its genome, not all those mistakes are tolerated. And, so you can experimentally demonstrate this in the lab. Like I worked next door to a lab that worked on an RNA virus called norovirus, which causes, it's a seasonal sickness virus. It's a diarrhea virus. And so what they would do is they would make mutations in the viral genome, and then they would infect cells of the virus, and they'd recapture it later on and sequence it. And what they found was that the majority of the mutations they made were reverting to the original version of the sequence when they took it out. And the reason is because the mutations they were making were deleterious on what that virus was able to do inside the cells. So were selected outside the gene pool. So the kinds of changes you see in viruses over time, only some of them will be tolerated in the bigger picture of that viral population. So in terms of co-infection, I mean, you can kind of see that really, though, with viruses like influenza would be a better example, because influenza has a genome of 8 segments. It's a segmented genome, so it's 8 parts to it. And so what happens is you can have 2 different types of influenza infect the same cell. And the reason why influenza is so dangerous is because you get what's called antigenic shift, and they can swap gene segments between them. And so you can have 2 fairly benign variants of influenza virus, and you can have a reshuffling of those segments and suddenly come out with a virus that's extremely dangerous. And that's one of the reasons why it evolves. It can just have this big jump in pathogenicity because of this swapping of genes between 2 serotypes of influenza within the same cell. So that does happen in that case.
So within the same infected person, you could have what, recombination of 2 different types of influenza to make one that would be, far worse, far more pathogenic, let's say?
Absolutely, yeah. I mean, that's, again, it's a well-known phenomenon with influenza, again, called antigenic shift. So there's 2 different types of evolution.
Since virions are non-motile, then this happens inside a cell, but that seems to say that the one virus, one cell model is not accurate in this case. That means that multiple influenza virus or virions will infect the same cell.
Absolutely. Yeah, I mean, absolutely. I mean, experimentally, so when you're working with viruses in the lab, you're typically not infecting. It's what they call MOI or multiplicity of infection. And so you're typically infecting with an MOI of 10 or 100, viruses per cell. I mean, you calculate it, you teeter the virus and you infect cells. One virus doesn't necessarily get a foothold. You need several viruses to be able to do it. So in a normal infection in the body, you're probably having multiple viruses per cell anyway. So if you have a situation like that with a mixed population of serotypes of virus, you're probably having that happening all the time, depending on the percentage of those serotypes within the population. I mean, it's with influenza, it's a much better characterized phenomenon than it is with something like SARS-CoV-2, because obviously SARS-CoV-2 is very new and early, and all those studies haven't really been done.
Well, another possibility, though, is that, once a virus has entered a cell, as it's, producing more virus and packaging it up in virions, that there's errors that occur, and then these errors eventually produce a more virulent form, or the different strands of RNA or DNA then recombine inside the cell from the same original father virus, and therefore are different.
Yeah, no, absolutely, it's entirely possible. I mean, taking sort of the macrocosm of it, though, and just sequencing enough virus over a broad population demonstrates, though, with SARS-CoV-2, this isn't, certainly if it is happening commonly, which you would suspect it is with an RNA virus like SARS-CoV-2, and it's a 30,000 base pair RNA genome. So the error rate for copying RNA is about 1 in 1,000 base pairs every single time it's copied. So those mistakes are happening even every single time the genome is copied. But the issue would be is the overwhelming majority of those mistakes are deleterious on that virus's immediate survival after it starts producing a virion and leaving the cell. And that would be, I would guess, that's probably the reason why those mutations aren't maintained. Now, it just so happens to be that eventually you're going to have a mutation which gives it some sort of preferential advantage and makes it worse. I mean, that's entirely possible. Yeah.
So if multiple viruses can infect a cell, do you think there's any coordination in the fusing portion of it, or the binding portion? Do you think that, if you imagine a cell and let's say 2 viruses find receptors that are, 50 nanometers away, let's say, and they both fuse and bind, and do you think that they could, for instance, maybe use the cell membrane or the cell internal machinery to coordinate action between them? Do you think that might be a possibility?
I think, again, being quite selfish quasi-life that they are, if anything, they're competing with each other to get inside the cell. I mean, there's only a certain number of cognate receptors in the surface of the cell. So I'm not aware of any cooperativity in entry that I'm familiar with. But I would say, the more virus, if you've got multiple strains of competing virus, every single one that binds to the cell and gets inside, it reduces the possibility of a secondary infection. Because again, remember, when the virus binds the receptor and the virus are taken inside, because it's so it's an endocytosis process. The whole receptor and the virus are taken inside at the same time. And so, well, not in all cases, but certainly for receptor-mediated endocytosis, that's how it works. And so that's one less receptor for future virus to bind to. So I would say more so it's actually reducing the chances of other viruses binding. And the other factor is that once one virus gets inside the cell and it, say, triggers interferon or triggers in a danger state, that's making it even more difficult, not only for other viruses to get into that cell, but also adjacent cells. So that's probably more the case, I would imagine.
So do you think that viruses are, if I imagine a virus as like a person sitting in a control room, like in a nuclear reactor, he's got all these controls in front of him. When a virus enters a cell, it obviously it co-ops cellular machinery to make more of itself. It modulates the immune response, et cetera. Do you think that viruses are capable of changing cells so that they'll send out specific extracellular vesicles that can send messages to other cells and change them? Do you think that's possible?
The extracellular vesicle issues, it's a fairly new area, and I mean it's not something I've worked on myself. I'm not familiar with anything virus related in extracellular vesicles. There's one thing that sort of touches on the stuff that I've been doing. I mean, one of the secondary messengers that's triggered by viral infection. So I mentioned that cGAP protein that detects viral DNA, for example. That produces a secondary messenger called cGAP. And cGAP can basically travel between cells and extracellular vesicles. It can also package inside virus capsids. And so when the virus picks up this danger signal and carries it to other cells and pre-warns the cell that way as well, there's been recorded instances of that. But in terms of the virus modulating the cell to send messages to other cells, I'm not too familiar with that. I mean, there's plenty of examples of vaccinia virus has, a good example of vaccinia virus modulating the surrounding environment. It's basically stolen the extracellular parts of loads of these pro-inflammatory cytokine receptors. So it's stolen the outside part of the TNF receptor, the IL-1 receptor, the interferon receptors. And what it does is it produces them inside the cell and makes it secrete them. And so the entire extracellular environment gets flooded with these soluble versions of the receptors that are needed to respond to the cytokines that the cell is trying to produce to get rid of the virus. So vaccinia virus would be a great example of this sort of nefarious, mastermind inside the cell modulating the extracellular environment for its benefit.
Yeah, that's amazing. Huh. And how could a naked piece of genetic material do all this? It's incredible.
But it does it by having a very, very large genome and an awful lot of time to come up with these ingenious ways of getting around things.
Well, some viruses, though, have a very small genome, right? Like I guess Ebola and some of those are really, really, there's not much going on, it looks like,
No, absolutely. So, I mean, there's a huge difference between DNA viruses like poxvirus. I mean, poxviral genomes are hundreds of thousands of base pairs in size. And I mean, they don't have to worry about space nearly as much as RNA viruses. They're only a couple of thousand base pairs long, But what I will say is what's been invariably found is, even though there's only a handful of proteins produced by these RNA viral genomes, they pack in a huge amount of multifunctionality. So you often find that you'll find an inhibitor that targets, say, interferon signaling. And you'll find that it also targets about 3 other pathways. And how they do this is quite remarkable. It's a kind of a wonder that they're able to pack so much functionality into a single protein by, again, refining it over time to add all these extra functions so that you only have to pack it into a small bit of genetic information within the very, very, very small RNA genome.
If I get infected with a virus and I label myself number 1, let's say I'm the first person infected, and then I give it to you and you're number 2, and you give it to your cousin, he's number 3, and by the time we get to like number 20, it's passage through like 20 people. Mm-hmm. What do you expect the virus to look like? How will it have changed? Will it be more commensal, more virulent? Will there not be much difference? What would you expect to see in terms of the outcome of the person that gets it and the virus itself?
I suspect it doesn't change. It depends on the pressure, because there's got to be a selection pressure to a great extent. So there's got to be some sort of selective pressure to maintain the mutations that happen in say, a small RNA virus. And again, going back to that population sequencing of this virus over and how many people it's infected now, it's probably in the order of hundreds of millions, if not billions, probably is going to be the final number when it's all said and done. But the fact that it hasn't changed substantially in so many people indicates that there's no selective pressure for those mutations that are probably regularly happening to be maintained. Now, that being said, viruses can change given enough selective pressure. Viruses are the most deadly when they've just jumped from other species, by the way. And so you look at this virus, it's just come from bats, probably bats, more than likely. And they're at their most dangerous because they're very, very poor at switching off the systems in humans, and that's one of the reasons why it causes such a profound inflammation, because it's not able to get around it. Now, you would expect over time that it will evolve gradually to be less dangerous, which is what most viruses are after, is essentially just producing as much virus as possible, flying under the radar and not making the host sick, because sick hosts can't move around and they can't transmit the virus. And dead hosts can't transmit any virus. So it's not the agenda of any virus to kill a host. It usually is the case that it's a virus that's just recently jumped a species barrier and now finds itself in an environment that it can't control and orchestrate. So you would expect over time it will change for sure. But what's more likely going to change is that when most people develop immunity to it either by directly getting it by vaccination, that any subsequent versions of it probably won't be dangerous anyway, because it'll be cross-protected by having antibodies. It's one of the reasons, most common colds in winter are coronaviruses, and not that dissimilar from this one. But they're different enough to the extent they just don't kill you, because they have adapted to humans to the extent they don't cause a serious disease. And they probably jumped from another species, just like SARS-CoV-2 has. But over time, they've become less dangerous as well. So it's a process of adaptation that all viruses are sort of evolving towards in order to maximize replication and maximize spread.
Well, very good. Well, Gareth, it's great talking to you. You have tons of knowledge. Definitely want to have you back to talk about your research. And then what's the best way for now for people to find out more about your research and your work?
There's a website on the Trinity College Dublin page. I very infrequently use social media, to be honest. I've got a Twitter page, but I very infrequently update news about SARS-CoV-2, but I wouldn't be probably a great person to follow as a result. But yeah.
Okay. Well, no problem. Well, Gareth, thank you for coming on the podcast. I appreciate it.
You're very welcome. Very welcome.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.